Okay, assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, everyone. Right now it's 8:20. Okay, and we are going to do something a bit different today. We have an exam, upcoming exam in like less than 2 hours, and you know, we're going to do a revision for it. Haven't touched the subject in a while, so I will just go through some exam papers and hopefully we will pass the exam. Bismillah. We're going to study for like, I think an hour 15 minutes. Okay. Okay. Setting the Pomodoro timer for 60 minutes. Come on, say it. There we go. 60 minutes. Starting now. Allah. Just go back here and then this over here. Let's go. What the hell is this? I don't remember it being like this. Okay. Let's just go quickly with so I will just try to answer them out loud. Okay, so multiple choice. The most conspicuous organelle in a eukaryotic cell marks the major difference from prokaryotic is the nuclear cells, obviously. The nuclear envelope is formed from two distinct lipid bilayers. Can we use it? We can use it. There we go. So this is B. The outer membrane is continuous with the membrane of the rough ER. The mesh network of intermediate filaments providing support to the inner membrane. The meshwork of intermediate, it's called the nuclear lamina. To the inner membrane, nuclear lamina or nuclear matrix. It's the lamina, B. The lamina is probably composed of lamins. The fluid model, the fish trap. The model commonly accepted for the nuclear pore complex structure is the fish trap. Fish trap model. Okay. The part of the nuclear pore complex that faces the cytoplasm, it's the cytoplasmic ring. Yeah. The primary function of the nuclear pore complex is still as a bidirectional dual functional hydrophilic channel. How is it? Why is it bidirectional? Because things goes in and things can be exported and imported. How is it dual functional? That's the question. I have a written somewhere over here. Yeah, bidirectional because it have dual function because it is active, passive and active, and a hydrophilic channel because it's a hydrophilic channel. Yeah. Okay. Now we have the 9th question. Chromatin is mainly composed of DNA and RNA, DNA and histones. That's B. So, yeah. Okay, next question. Page down. Okay. Not very useful, but okay. Anyways, we continue. So, the basic unit of chromatin fiber, it's chromosome, or it's a nucleosome. Okay. Each nucleosome core protein consists of a histone core, approximately 146 base pairs. The histone that reside outside is H1. The chromatin that is diffuse state is in diffuse state and during interphase and is hypochromatic is called euchromatin. Yeah, I'll double check. Yeah. Yeah, it's euchromatin. Permanently compact that is never transcribed is constitutive heterochromatin. The bar body is an example of facultative heterochromatin. The structure that rivets the two chromatids together is the sister chromatids. Yeah, it's the centromere. The region of chromosome that contains rRNA genes and form nucleolus are the NOR, nucleolus organizing regions. Okay. Exert an important role in maintaining entirety of chromosomes. It should be integrity. Yeah, integrity of chromosomes. So it is regarded as a timer of cell division. That's the telomere. The enzyme responsible for synthesizing telomeres is telomerase. According to the Denver system, healthy male, so there should be an XY. Looks like 46 chromosome. Alright, if anyone is wondering what I'm having right now, it's oatmeals along with some coffee. Chromosome banding is helpful for this, analyzing subtle characteristics of chromosome. The nucleolus is composed of 3, which is not of the, it's the laminar, nuclear lamina. So the active region of RNA transcription within the nucleolus is the dense fibrillar component. Pretty sure it is. Yeah, it's a dense fibrillar component. Active close to the border of the FC. Okay, so let me find the paper, exam paper that I had. This one is definitely one of them. Not one of them. It's big A2 paper. Yeah. Really easy to find. This is the only one that I have in my disposal at the moment. As the rest, I'm not sure. They're on the sheet, but yeah. Okay. Okay. Check out the papers. This is not this paper, but anyway, it could be helpful as well for later. Anyways, going on. And just double check so you don't get the wrong answer. Yeah, my answer is correct. Going on. The granular component, the nucleolus disappears during the phase of the M phase. Yeah. The nuclear matrix is mainly composed of proteins. According to the presence or absence of nucleus, cells can be divided into prokaryotes and eukaryotes. The nucleus is also called a interphase nucleus because by this phase. The outer nuclear membrane is studded with ribosomes. The black is a mesh of network intermediate of the inner nuclear, the nuclear lamina. The disassembly of the nuclear lamina prior to cell is induced by the water flammens. By the, I don't remember. By the this assembly of the nuclear lamina prior to cell division is induced by the lamins. Let's see. What's the answer? Phosphorylation. Are synthesized in the cytoplasm and imported in the nucleus. Karyophilic proteins. The main components of chromatin are DNA and histones, like we answered here earlier. Where is it? Yeah, DNA and histones. The DNA double strands are held together by hydrogen bonds between the bases. The nucleosome core particle is wounded by 146 base pairs of DNA. Histone is the linker histone that connects the nucleosome core particles. Histone 1. Histone 1, yeah. In female mammalians is the bar body. The constriction were central in the primary constriction. The activity of has positive telomerase ligands and describe the structure of the nuclear pore complex. You can say we can write this down. If you're written questions, I'll just write them down. We can just answer it ourselves instead of depending on, you know, AI. So you could say it's it has fish trap model. Models similar structure, or its structure is similar to the fish trap model. It has 2 rings. One facing the cytoplasmic, one facing the cytoplasm, called the cytoplasmic ring, and the other facing the nuclear, which is called the nuclear. Okay, it has a central plug. Actually, before the central plug, we should mentioned that 8 short filaments that are on the, you know, cytoplasmic ring. 8 long filaments or 8 and the 8 short filaments or granular filaments that act as receptors. So the filaments or the short filaments or the granules that are on the cytoplasmic ring, they act as receptors, and there are 8, 8 of them. And on the nuclear ring there are also 8 but long filaments, and there's a center plug alongside with it that is held by spokes. Okay, that's it. For the nuclear complex. Explain the structure of a nucleosome and its role in chromatin packaging. So we can say, the nucleosome is it's composed of 8 units. 2A, 2A, 2B, 2B, and then 3, 3, and 4, 4. Okay. So the DNA wraps around this 3D cube made out of 8 subunits, and we make, a nucleosome by it, and it has 146 base pairs, and it's its role in chromatin packaging because it allows us to to coil up the DNA in a manner where long DNA codons, length could be stored without having any issues, without having taking too much space. So it is like an efficient way to package the DNA coils. I'll just leave that to that. Okay, compare and contrast euchromatin and heterochromatin. Okay, euchromatin. Let me see if I can find it over here. Let me just show these as well. Okay. So the center plug is also called a transporter. Import carrier protein كده RNA كده. So A, to provide nucleus or relatively. What's the function of the nuclear envelope? So to provide nucleus or relatively independent environment. Translation and transcription can work simultaneously. And B, to control the material exchange between the cytoplasm and the nucleus, such as enzymes. Enzymes are made in the nucleus, are made in the cytoplasm, as ribosomes are made in the nucleus. But enzymes, such as enzymes are made in the cytoplasm and function in the nucleons, so they must be transported into the nucleus. And C, are taking the location and separation of genetic material. Every 6 nucleosomes arises to a 1 spiral to form a solenoid. So, for a chromatin to be functional, it needs 3 elements, which are centromere DNA sequence, telomere DNA sequence, and autonomously replicating DNA sequence. Centromere, telomerase, and autonomously replicating. Metacentric, submetacentric, acrometacentric, and telecentric. So the function of the nucleus is to, firstly, it is the place where, you know, our RNA transcription takes place, and it is where, you know, ribosomes precursors are assembled. So to sublitocentric. That's it for the nucleus. Now we go to the do this thing. What is it? Go to the review. We have like less than 20 minutes left. 17 minutes to be exact. Write this down real quick so we get it out into our quick access memory, random access memory, you know. The central dogma is a core. Molecular biology principle that states that describes genetic material flow. Meaning DNA to RNA, RNA to protein. Facilitated diffusion, passive transport, where molecules. No, this is facilitated, yeah. No issues with that. Enzyme blending receptors. Cell junctions consist of multi-protein complex. That provide contact A group, okay. Now, in the membrane system, a group of membranes and organelles in a eukaryotic cell that work together to modify, package, and transport glucose. A group of cell organelles, a group of membranes and cell organelles in that eukaryotic cell that work together to modify, package, and transport glucose. It's a functional, not a structure, meaning components are interconnected by a physical or transfer membrane between segments between them. It's a functional, not a structural. Most voltage gated and ligand gated. Back to EAG and IP3. And those are in the intermembrane system. Yeah. No. It's in the Krebs cycles and whatnot. Anyways, here's cell signaling. Anyways, we'll continue. how we do the we just want to write down so sized uni/multicellular membranous DNA circular DNA form ribosomes size cytoskeleton cell division type and then production. So, to sum up the differences and to categorize the differences between the prokaryotic and eukaryotic cells, we, have them in categories. In how many categories? 1, 2, 3, 4, 5, 6, 7, 8. 8 categories. Size, and whether they're unicellular or multicellular, membranous organelles, and nucleus, and DNA, ribosome size, cytoskeleton, cell division, and reproduction. So size, multicellular, membranous, DNA type, ribosomes, cytoskeleton, and what do you call it? The two last things that are closely related is the cell division and reproduction. Cell-induced cell receptor mediated endocytosis. So, ligand binds to the receptor, and then lateral diffusion happens. Then imagination, formation of a vesicle, and then coated and uncoated. Then class 3. Fusion with the early endosome and release of a ligand, so it can be So let's put it simply. Ligand bind to receptor. Then lateral diffusion. Then imagination. invagination, and then vesicle formation. Then vesicle, and it's coated, so we'll do uncoating. Then fusion with early endosome. Which can go into 3 ways. Late transport to late endosome. Good. Transport to late endosome, and then transport of the receptor to cell surface for recycling, and then transcytosis. Okay, draw the anchoring junction of the mnemonic. Boy, oh boy. The exam is in less than an hour, and then I have to, just review. Hopefully everything will go by smoothly, inshallah. Okay, let's see. Draw the construction of an anchoring junction and then remark. So I'm doing out the transmembrane linker. Pew. Pew, pew. Pew, 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 pew. Then pew. Pew, 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 pew. And then out there. So those are intracellular attachment proteins, and those are transmembrane linker proteins. An hour passed away. Okay, let's see. Let's study. How many minutes do we have left? We got 40 minutes. Let's study for 15 minutes more. Okay, let's go through this slowly. So the function of the smoothie ion is the detoxification, storing of calcium ions, and lipid synthesis. The addition of products from licensing can be used to rectify it. License all designs made in the rough ER. They move to the 𝐒𝐭𝐿𝐲 apparatus for modification. And the cis, this is 𝐒𝐭𝐿𝐲, a phosphate group is added to the monos, making M6P. M6P, acts as an address label, saying that send this enzyme to the lysosome. So in the trans 𝐒𝐭𝐿𝐲, they are, in the enzymes or the proteins enzymes, they get attached to the receptor, and then the receptor causes the receptor mediated endocytosis, and it's kind of similar to endocytosis, and then it forms a vesicle, and this vesicle goes to the lysosome. And because of the acidic nature of the lysosome, of the endosome, it deattaches, the receptors deattaches from the endosome. Okay. And because of that, the receptors go back to the 𝐒𝐿𝐲, TGN, trans 𝐒𝐿𝐲, trans phase, mature phase. Okay, that's about it. Based on the condition of the 𝐒𝐭𝐿𝐲 contains loose bacteria dispersed throughout the nucleus. Appears lighter under a microscope due to its low density, genetically active. This open structure allows transcription machinery like RNA polymerase, access DNA, enabling gene expression. It represents majority of the transcriptionally active DNA. Heterochromatin. The state of the chromatin is highly condensed and tightly packed throughout the cell cycle. and it appears darkly stained under the microscope because of its high density. Activity: genetically inactive, and the type packing prevents gene transcription by blocking access to the DNA. It's probably found in the centromeric and telomeric regions. Okay, that's about it. Yeah, I think that's enough. Come on, head out now. Yeah, it was a good revision. 1 hour and 15 minutes. Okay, hopefully we, pass the exams or get a very good grade at least, and yeah, I'll come back later and hopefully continue the anatomy journey. We will continue with the, let me check. With the sphenoid and with the occipital, sphenoid and ethmoid. Actually, occipital and then occipital with ethmoid and then the sphenoid. Okay, I'll be on my way now. Hope you guys learned something. Then what? But yeah, anyways, I wonder how would I see this after like 5 years from now, 10 years from now even. We'll see. Anyways, دمتم في أمان الله.